we're going to go a bit experimental today. We've not got one speaker, we've not got two speakers, we've got three fantastic speakers. They're all PhD students, they're up and coming mycologists, they are the future of mycology, but they're here now and they're amazing. So the form we're going to have tonight, we're going to give each speaker 20 minutes to give a talk. There'll be 10 minutes for questions, and they'll go on to the next speaker and so on. And there'll be some space for open questions at the end. As always, any questions you have, please put them in the chat. And I'll just, as we come to a period of questions. But other than that, I'll stop there and I'll pass on to our first speaker, Chris Cook from NIAB Cranfield. Chris, do you want to unmute and give your presentation?
Thank you very much.
Welcome.
Okay, so hopefully you can all see my screen okay, and everything's working well. So firstly, thank you very much for coming. It's good to see so many people here, and hopefully you're interested in what I can tell you about today in terms of my work. It's an element of mycology, but it's a bit more broad in terms of ecology and soil science. So hopefully there'll be some good points in here that you can all take. So I'm going to be talking about apple replant disease, a positional and genetic strategy for disease management. And as mentioned, I'm based at NIAB in Kent, partnered with Cranfield University in Bedfordshire. So, oops, slide changes. So my background, I studied biology at undergraduate level at the University of Kent, and then started my PhD in 2018 on the CTP FCR program, which is an industry-led PhD program, recently started up in the last few years. And I'm part of the pathology group in the pest and pathogen ecology department at NIAB now. And sort of my overall project title is Understanding Resilience of Soil Beneficials to Combat Apple Replant Disease. So when I'm talking about replant disease, probably best to give it a bit of context. So what do we mean by apple replant disease? Well, it's defined as the planting of successive generations of the same crop without rotation or land rest, and you get a reduction in the overall performance of the tree after you replant the new generation of tree. It doesn't just affect apples, it can affect members of the rosacea species and other top fruit as well. It's been observed in over time. And it's more prominent in dessert orchards just due to the nature of how the systems work. So it generally affects that orchard more because it has a quicker turnover than a cider orchard that will be in the ground for 40, 50 years or upwards, depending on the performance of their orchard. So the symptoms of replant disease is stunted growth of the tree, a reduction in the overall vigor of the tree. Obviously, you get lower yields with a lower vigor of tree, and underground you get root tip necrosis and a general reduction in root health. Sort of these two images on the right-hand side here give a good indication of sort of the above ground effect. So these are the two identical trees at planting. One is suffering from replant and one is healthy. And at the top is sort of the below ground effect. So replant soil on the right hand side, and on the left hand side is a healthy root system. So what causes replant disease? It's a biotic disorder. So it's caused by a complex of pathogens. And you basically don't need all of the pathogens present for the disease to happen. Oh, sorry, I'm getting some background noise. You don't need all of the diseases to be, the pathogens to be present for disease to happen, but generally it's a complex of a number of them does cause the disease. So there's two major sort of areas, which is the oomycetes, so pythiums and phytophthora are most commonly associated with replant disease, and fungal pathogens, the cylindrocarbon species, Rhizoctonia and fusariums, various fusariums also implicated with replant disease. So quite a broad spectrum of pathogens implicated with it. And it can be worsened by root lesion nematodes. So obviously the nematodes make lesions in the root system and allow for the pathogens to enter into the root and infect through those wounds. So current treatments for replant disease, the major one and the most effective one is chemical fumigation. So applying a broad spectrum fumigant onto the entirety of your orchard. This is obviously expensive, which is the most important problem with it, but there's also pressure from government legislation banning most effective fumigants we have. And any fumigants we can still use aren't very effective at treating the pathogens that I mentioned previously. Another method is anaerobic soil disinfestation. So this involves adding a product and covering your soils with plastic for a period of time to create anaerobic conditions in your soil. And what they believe this leads to is a greater number of bacteria in the soil, and then they in turn control soil-borne diseases that may want to invade those spaces by niche exclusion. There's soil biofumigation, so trying to use biological products to achieve a similar effect to what the chemicals were doing. These are things like brassica seed meal that's been extensively studied in the USA. These have an antifungal and anti-nematode action, but they're ineffective against oomycetes. So you control one side of it, but there is still the potential for phytophthora infection, for example, which alone is a bad disease. So it's not a massively viable candidate long term. The other way to look at it is, can we apply beneficial products to the tree to improve the health of the tree to overcome replant disease? So this would be the addition of products like arbuscular mycorrhizal fungi that work symbiotically with the root to exchange nutrients and produce a beneficial effect. Biocontrols, so anything that will control the pathogen complexes in the soil. Biological products, of course, or plant growth promoting rhizobacteria that have a similar effect to arbuscular mycorrhizal fungi, but obviously from the bacteria side of things. There's also cultural practices that are important with replant disease. So the planting position, you want to try and find soil that is as close to virgin land as you can. That's pretty hard in an old established orchard, which may or may not have, for example, irrigation lines already put into the orchard. So the best thing would be to plant in the alleyways next to the previous tree station, and that's something I'll touch on later in this presentation. Obviously, if you can move your entire orchard, fantastic for you. But again, that's sometimes not the best method, because even in nurseries where they have a rotation around different fields around where their land, 20 years between rotations when they put apples in, and the next time apples go in that same land, they see a replant effect. So it's not the best way to do it. You can also do a break or cover crop. Obviously, cover cropping can produce beneficial effects on the soil, or a break crop, but if they're taking apples out of the ground, then that's eating into the profits of the grower. So this all comes down to economics in the end, and if a grower has to take his apples out of the ground, he's probably not going to do that. He'd much rather treat the trees while they're there. Good hygiene: if you remove a tree, make sure you remove all of the root system that's in the ground, because it might be carrying the disease within the roots. But again, that's difficult. You might want to get it out quick, or you use machinery, so you can't go through with a fine toothpick to identify any root that might be left over. Or we can look for genetic tolerance, or genetic relationship between rootstocks. And again, this is something I'll touch on, but different rootstocks will have different tolerance to replant disease. So when you're selecting the genotype for your rootstock, so the rootstock is obviously the part of the tree that's under the ground that dictates how large the tree will be, and then the scion that you graft on the top will dictate what fruit that you have. So when we're looking at a dessert orchard, you want a smaller tree, sort of the size of an average person, so they can pick most of the fruit by hand from the top of the tree without any issues. In a cider system, you want the tree to grow as large and as bushy as you can, so when you apply chemicals to your tree or whatever, sprays, you're hitting a large area, and when it comes down to harvest, you let the apples naturally fall off the tree, so there's no need for picking. So you'll tend to pick a more vigorous, larger tree for a cider system, and a smaller, less vigorous tree for a dessert system. But what we see when we look at, these are just some examples of some different rootstocks that are generally used and their parentage. The most dwarfing trees compared to the most vigorous trees, the relationship between the tolerance to replant disease isn't linear. So you might get some very dwarfing trees that are very tolerant, and some genuinely more vigorous trees are more tolerant because they're bigger, but that relationship isn't linear. So you can't just always go, I'm going to plant a bigger tree and it will be more healthy through replant. Studies have shown that the Geneva rootstocks, so the G series, are more tolerant than the Morning rootstocks or the M series. But again, they're different trees, so you'll have to take into account how they're going to look in your orchard. And they believe that the difference here is that Geneva recruit a slightly different bacterial rhizosphere than the Morning do in terms of their rhizosphere populations, which might lead to the tolerance to replant. So I want to talk a bit about what I've done to do with this now. So we had a 5-year long trial part of the UKRI Horticulture and Potato Initiative. It was ran in a Heineken orchard. You've probably all heard of Heineken, make Strongbow cider, based out in Worcestershire. And we planted 7 different rootstock genotypes, sort of the most commonly used in the industry. And we grafted them onto the variety Worcester Pear Main, which was a good candidate because it can be used as a cider apple and as a dessert apple. And we planted the trees in the previous tree station where the old trees were, and in the alleyways between, so that the rows were much closer than you would have in a normal cider orchard. They were previously planted onto Katie apples, so if you've ever been to the cider section and seen Thatchers, you'll know how important Katie is to the industry. It's one of the most commonly used cider varieties of apple. And these were on MM106 and MM111 rootstocks before, so quite vigorous rootstocks. So what we theorized here is if we saw an increased establishment in the alleyway compared to the previous tree station, there's a replant effect occurring on the trees in the tree station. If that makes sense. So to measure this, we checked the height of the tree, the girth of the tree, which is probably our most important measurement. To standardize that, we did it 5 centimeters above the graft union. We then collected soil rhizosphere, extracted DNA and sequenced the 16S and ITS regions to identify the bacterial and fungal populations in our soil respectively. We then generated operational taxonomic units. So that's a big word, but basically an OTU is just a simulated species generated through sequencing. And we clustered those at 95% similarity between sequences to try and have some, so they're quite different. And then what you can do is assign those back using taxonomic databases to have a simulated species that you think that OTU relates to. We then calculated diversity indexes to see if there was any difference in the fungal and bacterial populations we had within those soils between the tree station and in the alleyway, and between genotypes as well. So some results from this. So this was basically the girth data that we had. And simply what we did was we took the difference between the girth from the alleyway to the tree station. And if that value was positive, then the value would be above 0 in our model. We used a model that just indicates the slope, so the difference over time. And as it becomes more positive, so the difference is larger between the alleyway and the tree station, this value of the slope will go a lot higher. So anything we saw above 0, we theorized had a replant effect, and the higher it goes above 0, the more severe the replant was. If it went below 0, we would see the inverse effect, and it was actually going better in the tree station. I mean, normally that wouldn't happen. Sometimes you might see something quite close to 0, but generally not something very, very negative. So what we saw, interestingly, was 5 out of the 7 rootstocks that we looked at on the site had some kind of replant effect going on. 5 of them were above 0 here. And the most affected, so the most severe, were 116, 106, and M200 on this site. So now I assigned a score to these, so 2 being the most severe and 1 being the intermediates in the middle here, the 2 in green and purple, and then 0 was no replant effect, those ones that were below 0. So what makes this interesting is that what we saw was 116 and 106 were the most effective. But when we look at this chart of vigorous to tolerant, we see that 116 and 106 should be quite tolerant. But what we know is that previously planted on that orchard was 106. So what we see is actually the same rootstock that was planted there previously has a replant effect. And the offspring, so that one of the parents was 106 here of 116. So the offspring of that genotype also has a replant effect. So it's quite interesting that there may be a genetic link that we previously weren't sure about. We thought it was just due to pathogen complex, but there might be a genetic link between how those pathogens end up in our soil or some different factors there that means we get more severe replant disease. So when we look at diversity, this again is quite complicated, but I'll try and simplify it. So we have the 0 score, so no replant. We have 1, which is intermediate, and 2, which is most severe. And then it's colored, the shape, sorry, is the alleyway in circles and tree station in triangles. So you want to try and see a difference here. And alpha diversity is quite a simple metric. It just takes into account the overall diversity that's there. So if you had a pond that had 5 fish in, it would be a diversity of 5. But if you had a pond that had a completely different 5 fish in, it would still be 5. So even though they have different diversity. This doesn't take into account that. So you have to take this with a little bit of a pinch of salt, but it's a good starting measure here. So we see a slight reduction in the alpha diversity between the alley and the tree station, which is interesting. So maybe in the most severely affected, there might be an effect of there might be a reduction in the bacteria here. And similarly for fungi, we see this effect, but genuinely it's quite similar across treatments, and there's no real difference between those severely affected by replant and those not in terms of alpha diversity. But when we move on to beta diversity, so this takes into account the differences between species. So if we had a pond that had 2 similar species to another pond that had 2 similar species, then beta diversity would have a number of 2. So it takes into account actually those that are different rather than the overall how many are there that are different, if you know what I mean. So it actually compares the 2. And what we can do is we can put this into a 2D format. So I'm using non-metric multi-dimensional scaling here. So it's just a way to condense a lot of information from, we have a lot of OTUs obviously, because there's a lot of different species in the soil. And then it puts it into a 2D format. So this is the location, so the alley versus the tree station, and we can see there's quite a big difference between the bacterial populations that we've got in the alleyway and in the tree station. So that's interesting. So what is it for fungi? We see a similar effect. There might be quite a big difference between the populations of fungi that we're seeing under an alleyway and under the tree station. When we're looking at sort of the differences in the replant, so the most severely affected and the less severely affected. So more specifically, the difference between the triangles and the circles here within a color, because we're most interested in the most severely affected. We see there's a big difference in terms of bacteria. So what we can theorize here is that bacterial populations within those replant affected trees are quite different. And when we look at fungi as well, it's less obvious, but we still see some kind of differences there. In terms of the populations that we see under the trees. So sadly, that's the point that this research is currently at. And my next step is to see what these are. I'm sure the questions are going to be, well, what are they then? Are they pathogens? Are they not? That's the next step, is to try and identify what taxonomy are actually different here, or significantly different to be more exact, because they can be slightly different and it won't have a huge effect. So that will be the next task. But so general conclusions from what I've said, because I know it's quite a lot, is the more closely related the rootstocks are, the more severe the replant disease was that we saw. So the 106 and its parent, it's the offspring of 106, 116. What we see is that bacterial and fungal populations in the rhizosphere are different when they're grown in the alley and the tree station. So that's quite interesting. Just a meter difference between where you planted previously has quite a big difference, but that could be due to compaction from tractors, or it could be due to any applications you put onto the bottom of the tree, fungicides, pesticides, you name it. It's probably been sprayed on there, especially in cider. We saw that the overall alpha diversity, so just a general how much bacteria is there, how much fungi is there, was similar between replant affected trees and non-replant affected trees, but those populations were different when we looked at beta diversity. Between the alleyway and the tree station, so the meter difference was quite significant, and that those most severely affected also had very different populations. So future work, as I said, would be to identify what those differences are. Are they pathogens? Are they beneficials? What are they doing in the soils? And try and find some candidates on this specific trial. So yeah, to summarize sort of the whole thing, how do we manage replant? So chemical fumigation is no longer realistic. Neither is virgin land, because they don't exist, and the chemicals now, there's too much pressure from government, and there's not enough chemicals around, and they're expensive to apply. So it's not a viable strategy anymore to deal with replant disease. More related rootstocks will suffer more. So at the very least, if a grower is replanting his orchard, he should use something genetically different. Obviously that comes with the drawbacks of, will it be the same vigor? And will it satisfy his requirements in terms of what type of orchard he's growing? Rootstock, so yeah, rootstock selection and planting positions are effective, but they're financially difficult. If you have pre-existing irrigation lines, etc., you're going to have to change all that, and that's a big financial burden to a grower. So chances are they won't want to do that, and they'll just end up replanting the same variety in the same spot, and you'll end up with a replant problem. So future replant research then should focus on genetic tolerance. We've identified that genetic effect, so it should screen whenever they do crosses and things in breeding to try and identify replant disease, tolerance genes, etc. And also the soil treatment. So if we can apply something that overcomes replant, then you don't have the problem of trying to select the best rootstock. So if that's possible, it would be a good strategy moving forward. So I hope that was all clear for everyone. I'm happy to take any questions that you have. And obviously, big thank you to everyone at Cranfield and NIAB who helped me out with this, and for you lot for listening, and the BMS for giving me the platform to speak to you. So thank you very much.
Wow, brilliant. What a fantastic talk. And as someone who likes apples and all their uses, one I found very informative. I imagine lots of other people did as well. We've actually got 4 questions in the chat at the moment, so we'll go through them. Yeah. If you don't get your question in time, don't worry, there's space time at the end, we'll go to the questions there. But the first question from Jane Haydecker asks, what plants can be used as cover crops?
So that would be things like brassicas, so name a brassica, like lettuces and broccolis and all sorts. They tend to not often be used for food, but there's people working on ones that can be used for that. But in terms of the financial output, it's difficult. But it tends to be like mustards and things. You know, you name them, like that sort of thing is the standard use for cover cropping. Those seem to get the best effect. And there are people working on using cover crops within alleyways and things. So it'd be interesting to see if that's a viable thing to grow, sort of double up, get your alleyway working as well as the tree station for fruit. So it'd be good.
We've got a question from Alex who asks, what methods of biological control have been trialed? Is there potential for mycoviruses to control the fungal pathogens such as Fusarium species?
Yes, so there's people working on that sort of technology for scab, but it's quite new and novel, but it's an interesting idea because they're so specific that they might be able to be used to target the individual pathogens. But the problem, like I mentioned before as well, is we don't really know what's massively causing replant. We have these candidate organisms, but we know they work in complex, but we also, the methods we use at the moment to identify what's causing replant is you have an infected tree and a non-infected tree, and if you find a difference, then it's immediately a candidate. But when you look in sort of the literature for what some of those are, they're actually not pathogens at all. So to try and find something that you know is definitely causing replant every time and then find the virus to do it. But yeah, it's amazing tech. Like I said, this the stuff for scab is very interesting, but to apply it into the soil as well would be quite challenging because you have one window when you plant and that's basically it. You can't really apply through the soil, but with a lot of the treatments like PGPRs, you can water them in and they tend to get down to the roots, so they might be a bit easier. If that answers your question.
Joni Wildman asks, how long has this disease been a problem and is it getting worse?
So it's been a problem for as long as papers have been published, pretty much. It used to be called SARS, specific apple replant disease. So you might have heard the term, not sure, like sardy soils. It's where we used to be, a lot of people would say, oh yeah, you know, I got sard in my soils and you know, apples aren't growing. But then some seasons, it's all down to environment as well. Some seasons are worse for replant and some seasons are better. But what the ones who really are saying it's getting worse are nurseries, but that's because they have such a fast turnover. I think it's getting to the point where they're having to abandon specific areas, or they're having to plant different rootstocks than what they were originally planting there because they just can't get the crops to grow and get the plants. They have like a 2-year window or whatever to make small rootstocks till they've got to send off to be grafted or whatever. So if that tree doesn't grow well in 2 years, then they're in trouble. And the thing with replant as well is it doesn't emerge for 2 years. Normally 1 to 2 years, once it's starting to fruit, once you're starting to see the good growth and fruit doesn't come off, and it comes off 2nd year growth on trees. So you don't actually see the impacts of replant disease until you put significant investment into the tree. So yeah, I'd say it is quite tough. Not in cider, as I said, so much, but they do complain about it. So it must be a fairly an issue because they don't tend to touch their trees much. And if they're mentioning it, it must be quite bad. But yeah, definitely in desserts, it's getting pretty bad. But we have a lot of new rootstocks coming out, so let's see how it goes with time, I guess.
Actually, following that question, I've got a question from Cameron Dicknig, who asks, why does ARD impact on newly planted trees, yet not do much harm to the previous planting? And he speculates, could it be that newly planted trees are not well established enough to resist?
Yeah, I mean, probably that's spot on. They're just weak, and they can't. They don't have the root system to be able to cope. They're putting a lot of their energy into establishing, building their root systems, things like that. A lot of stuff going on under the ground for young trees. So probably anything that's in your soil that's a pathogen is going to have a field day with new trees compared to old established trees. And they're going to have more, older trees are going to be able to move into different areas a bit better if there's a problem in one specific area, whereas you have to hope that sort of small cube of area that you plant your new tree into is good quality soil, I guess. That's why they tend to struggle so much.
Brilliant. We've got time for any more questions, one or two more questions if people want to ask one or two more questions. If not, Chris will be available at the end of the free talks. There's more questions for Chris if you want to double check on your apple trees or anything else beforehand. Okay, other than that, thank you ever so much, Chris. A really interesting talk. And I'm sure you have some more questions at the end.
Brilliant, thank you.
Next, we have Theresa Wacker from the MRC Centre for Medical Mycology in Exeter.
Thank you very much. 1 second, I'll try to share my screen. And that didn't work. So you should see it now. Can you see my screen?
Yep.
Awesome, many thanks. So first of all, thanks for having me. It's an honor to speak to you today about repetitive DNA and evolution of amphibian killer fungi. And before I start, just a couple words about myself. So this is me doing the thing I love aside from fungi and science. I'm a climber, and I come originally from Germany. This is also where I did my bachelor's in biology and my master in microbiology and biochemistry before I moved to Aberdeen to do my MRes in medical mycology and fungal immunology at the MRC Centre for Medical Mycology, which was back then still located in Aberdeen. It then moved to Exeter, and I moved with it. And I'm now doing my PhD there, which has the title Genetics and Epigenetics Underpinning a Broad Spectrum of Fungal Pathogens. Just briefly about the center. So it's located in Exeter, as I said, and has 3 major aims. First one being to improve the ability to detect fungal infections. Second one, to generate better, safer, and more effective antifungal drugs. And the third, to enhance the understanding of fungal virulence and host antifungal immunity, which is what my PhD is doing. I'm obviously not working there alone. There are many really amazing scientists working there as well. Amongst them, my first supervisor, Dr. Reese Farrer, and my second supervisor is Professor David Studholm, and he is in the biosciences, but not in the MRC Center. So let's get right into it. So what amphibian killer fungi am I actually talking about? So in the late 80s and early 90s, there were more and more reports coming of sudden mass mortalities of populations of amphibia in otherwise pristine and protected areas. And these were really enigmatic declines that several theories were put forward for the reason that might be. So for instance, UVB or weather or climate, but it took till 1998 to actually find out that the chytrid is causing this amphibian mortalities. In 1999, it was formally described as Batrachochytrium dendrobatidis. Until 2013, it was the only chytrid known to infect vertebrates, almost specifically amphibia. In 2013, a population of salamanders in the Netherlands was reduced to 4% of its original size within 3 years. And this is when Batrachochytrium salamander vorans was identified as the cause of that. So Batrachochytrids cause chytridiomycosis, and by now this is a panzootic. And panzootic being the equivalent of a pandemic, just in animals. And it's a panzootic because BD is now found on all continents. It's endemic in Asia, same as B cell, But B-cell is only found in Germany, in Belgium, in the Netherlands, and in Spain, and actually in captive amphibia in the UK as well. Chytrids are aerobic zoospore fungi, and they diverged really early, 750 million years ago. That's a lot earlier than, for instance, Ascomycota, the Basidiomycota, which diverged like 500 million years ago. Most chytrids are saprotrophs and parasites, but B-cell and BD are the only species known to infect vertebrates. And as such, they're responsible for sharp amphibian declines and extinctions. They're associated with 90 extinctions and 501 declines, of which 124 are more than 90% declines of the populations. So it's really a big problem for biodiversity, and it's contributing massively to Earth's sixth mass extinction. B-cell and BD have slightly different, so let me just get the point out real quick, have slightly different host specificities. So B-cell parasitizes caudaten species, that's salamanders and newts, and BD is a generalist. So it infects all orders of amphibia, even though it prefers endurance. B-cell causes multifocal superficial erosions and deep ulcerations in the skin, which you can see here in this poor little fella. And BD causes hyperplasia, which is cell proliferation, and hyperkeratosis, which is a thickening of the skin. And that's really problematic for amphibia because they actually do osmoregulation and respiration through the skin. So in the case of B-cell, the erosions and ulcerations cause a breakdown of the barrier function of the skin and cause sepsis in the host. In the case of BD, this hyperplasia and hyperkeratosis leads to a loss of function of the skin, so leading to osmoderegulation and ultimately cardiac arrest. While in B-cell, it has been observed that there's hyper-responsive immunity in hosts, meaning the host doesn't really mount any defenses. BD is hyper-responsive. And that doesn't necessarily mean that the infection is cleared, but the host shows some response. Mortality rates vary depending on host, environmental conditions, and pathogen loads, but they're really high with 87 to 100%. And when we look at the entire picture, like the many hosts and infects and how deadly it is, the question arises is how did the chytrids evolve to be such successful pathogens? And to look at that, we need to first take one step back and first look at repetitive DNA. So I don't know if everybody knows about repeats and transposons, so I'm just quickly introducing them, explaining them so we're all on the same page from here on forward. So repeats are part of the DNA sequence, a nucleotide sequence, which are repetitive, like for instance, here, CAG, CAG, CAG. There's many different kinds of repeats, and one specific kind of repeats are transposons. And transposons are also called jumping genes. So they can copy and paste or cut and paste themselves from DNA A to DNA B, thereby disrupting genes, duplicating genes, changing the regulation of genes. So they're really powerful players in a generation of genetic mutations. This is also summarized in this slide, which is a bit busy, so let me walk you through it. The most important thing to remember is that transposable elements cause mutations, recombinations, and rearrangements that create variability and adaptability. So they influence genome plasticity, for instance, genome size expansion, altering gene expression. They change the host range, have influence on pathogenicity, and they also impact evolution. And in this context, there is transposable elements are enriched in compartments in the two-speed genome. So what are two-speed genomes? Two-speed genomes have been found in fungal oomycete plant pathogens, like for instance, Phytophthora, Chris talked about. And what they are is basically they're bipartite genome architectures, where in one compartment of the genome, there's long intergenic distances and it's high evolvability. And the other part of the genome, we see short intergenic distances and genome stability. So these products of adaptive evolution show high evolvability, they are repeat rich, they're gene sparse, and they're enriched in transposable elements. On the other hand, we have the areas of genomic stability, which are repeat sparse, gene rich, and devoid or reduced in transposable elements. So this is a really elegant evolutionary solution to, on the one hand, have conservation, and on the other hand, have evolvability, so have genome plasticity, and also and also reducing the risk of excessive deleterious mutations. What that means is that these adaptable two-speed genomes confer a co-evolutionary advantage and allow for host jumps. So this graph is about plant pathogens, but it applies the same way to vertebrates. So when you have a pathogen with adaptable genomes, they are a lot less likely to get extinct, because they can actually, when the host lineage gets extinct or when the host becomes resistant, they can either overcome the resistance or jump to a new susceptible host, which is not possible for pathogens with a less adaptable genome, which are much more likely to get extinct. So this confers a big advantage for them in the co-evolutionary arms race. Now, this is all nice and well, but how do you actually investigate that? Like, how do you look at that? And what you need, first of all, is a high quality genome sequence. And we did have that for BD. We didn't have it for B cell. So what we did is we did some nanopore long read sequencing with this shiny machine, Promethean, on B cell. And the advantage of long reads is that you can resolve repeats. And we really want to resolve repeats. We want to see repeats because we're interested in it. I then did a genome assembly, which basically means I puzzle together from the sequence reads I get, the genome or a representative genome, and get a assembly of it, high quality assembly of it. And I then did analysis. I'm now going to present you some of my results, and I'm going to focus on B cell because of time. So when we got the new high quality genome assembly, what you could see first and foremost is that it's 3 times bigger than its closest relative BD. And in fact, of the 21 chytrids we looked at, it's the second biggest genome, with Onocladischitrium polystomum being bigger. Even though it's the second biggest, it has the most repeats. It has a whooping 40.9% of repeats. And this also reflects in the amount of transposable elements it has, 19.4%, which is a lot. And with BD having less than 5%. So this gives us a very good indication that B cell has undergone a large repeat-driven genome expansion. Now, we wanted to see if these elements are uniformly distributed. And actually, they're not. When you look at transposable elements, they are not uniformly distributed, but form islands. And I've marked a couple of them, not all of them. And that also gives us an indication or a hint towards maybe we actually do have these repeat rich and repeat sparse compartments. Before we get any further, I have to explain this graph to you because otherwise we'll all get lost. So what we look at here is a density plot. And each of these rectangles is a certain amount of genes. And you can see the color code here. And on the x-axis, we have the 3' downstream intergenic distances. On the y-axis, we have the 5' upstream intergenic distances. And when you look at this quadrant, we have both long upstream and downstream distances. So we have long intergenic distances, and we have a gene sparse region. So the genes that are fall into this area are in a gene sparse region. Conversely, when we look at this quadrant, we have short intergenic distances. We're in a gene-rich region, which we would associate with the area of genomic stability, while we would associate the gene sparse region with the area of evolvability. When we now look at B cell and its closest relatives, and we just look at all the genes, we can't see really an enrichment in either quadrant. However, when we look at different gene categories, we do. So for core conserved genes, which are housekeeping genes, genes who really, really don't want to mutate, they're found overwhelmingly in the gene-rich region, so the region of genomic stability. And this is actually significant, which is like indicated by these asterisks. When we look conversely at B cells M36 metalloprotease genes, which are prime candidates for virulence genes, and they're thought to be involved in the breaking down of the skin and the extracellular matrix, which caused these erosions that I showed you earlier. So they are found in the gene sparse region, the region of high plasticity. This is also true for genes with a secretion signal. And as you can imagine, when you have proteins being secreted, they're actually coming in contact with the host. So many effectors and virulence factors are actually secreted, and they are also found in the area of high genomic variability or evolvability. But most importantly, when you look at differentially expressed genes, so genes that are actually expressed in vivo during host infection. So when the chytrid is during the infection of the host, they are also found in the area with long intergenic distances, repeat rich, gene sparse, and highly evolvable. We can also look at that when we just look at log mean intergenic distances. And what you want to look at here is this rectangle. This is the median. So when we look at core conserved genes, we can see that they are a lot shorter, the log mean intergenic distances, than the ones of the virulence genes, for instance, M36s or the secreted genes. And this is a highly significant difference. It's less significant in BD, more significant in B cell. Also, when we look at the y-axis, which is the same for all of these species, we can see that the M36 metalloproteases in B cell have actually the longest intergenic distances of all of the species I show you here. So clearly M36s are important. They're candidates for virulence genes. They have been shown in dermatophytes to be important for host invasion, and they're largely expanded in B cell. So B cell has 177 of them, and they fall into 6 different families, which are color coded here. And the latest expansion, which is this red family, the latest expansion, interestingly, is associated with a line transposon upstream. So line transposons are retrotransposons, and that means they do a copy and paste mechanism and they can cause duplications. And so the latest expansion of these genes have a line transposon upstream and unknown repeat downstream. This is interesting because when we just briefly go back, we don't find that line transposon on any of the other older families, but just in the latest expansion. So that gives us, this leads to us thinking that this is actually linked to this expansion is linked to transposable elements, leading to the working model we have at the moment. So the line copy and paste machinery has duplicated M36 metalloprotease, leading to this huge genome family expansion of 177 metalloproteases, which contributes to the genome expansion and a huge size of the genome, and confers an evolutionary advantage on B cell, being advantageous in the arms race with the host. Taking it all together, and I'm actually far too fast here. Taking it all together, B cell has undergone a repeat-driven genome expansion, and it has a bipartite two-speed genome architecture, which we, for instance, also find in Phytophthora, to go back to what Chris was talking about. Putative virulence genes, like the M36 metalloproteases, which have been proven to be so important in other fungal pathogens, are enriched in genomic compartments that are the cradles of adaptive evolution. And M36 metalloproteases, the expansion of these proteases is linked to line transposable elements, which interestingly is the only family of transposable elements still active in humans. And for the most exciting thing for me personally, what I think is really cool, is the first time evidence that we find evidence for two-speed genome outside of plant pathogens in vertebrates, and I find that very exciting. This all wouldn't be possible without my lab with Dr. Theresa Wacker, who's my first supervisor, Nicola Salmstetter, who's our excellent technician who did all the lab work and did an amazing job for that, Dr. Hugh Gifford, who is the second PhD student in the lab, Dr. David Sutton, my second supervisor, Dr. Duncan Wilson, and Professor Matthew Fisher for their invaluable contributions, and in case of Professor Fisher, also for providing the chytrids. All of the MRC CMM and my funders, BSM, the British Mycological Society, for having me today. And most of all, you as the people listening, thank you so much for listening. And if you have any questions, fire away. Thank you.
Oh, fantastic. Thank you for a really wonderful talk, particularly someone who loves amphibians in all their forms. Very interesting. And we've got some questions jumping in already, so I'm just going to jump into them if that's okay.
That's okay. Thank you.
Brilliant. So we have Alex, who asks, is transposon proliferation associated bias linked with accessory chromosomes, and are accessory chromosomes linked with virulence genes of BD?
Accessory chromosomes are not necessarily linked to virulence genes with BD, but actually accessory chromosomes are one thing which we find in 2-speed genomes, and where virulence genes are harbored oftentimes. What was the first part of the question again? Sorry.
Is transposon proliferation associated bias linked to accessory chromosomes, and are accessory chromosomes linked to virulence?
It can be, but it is not necessarily associated with it, which is not really a satisfactory answer. But yeah, it can. But mostly transposable element proliferation has to do with because these transposable elements can jump into genes, which can cause deleterious mutations. Organisms have a machinery to actually stop transposable elements or to silence them. So, for instance, repeat-induced midpoint mutations, etc. So, when that isn't present, or when there is no selective pressure to not have these transposable elements proliferate, then they do. Yeah, I don't know if it answered the question. Sorry.
Thank you, sir. One answer. Cameron Dicknig asks, says, first of all, thank you for a brilliant talk. B-cell and BD are, in relative terms, modern diseases of amphibians.
Yes.
Where are they suspected to have originated from?
They originated from Asia, and in Asia, B-cell and BD are endemic. So they are found there in Apache distribution, but they're found, and they don't cause disease in the endemic amphibians there, because they have co-evolved over such a long time scale. So yeah, Asia. Short answer.
Thank you. And we've got a question from Alistair Brown, who says, fascinating talk, Theresa. The hypothesis that has been repeat-driven gene expansion is very convincing. But is there evidence for gene loss and contraction in repeat sparse genes, in the repeat sparse regions of the B-cell genome?
So if there is also loss of contraction of gene families and the repeat rich and the gene sparse regions, is that correct?
Yes, gene loss and contraction of repeat sparse regions, yeah.
Yeah, I haven't found any. So in my personal research, I haven't seen that yet. So no.
And just to ask, with this repeat-driven gene expansion, what is the prognosis for treatment of B-cell in the future?
So so this research specifically will not necessarily open new avenues to treatment. So for one thing, it's really hard to treat amphibian populations in the wild. And the other thing is, so it's basically, there's several things. So what we see is that we can't do vaccination, because repeated infection with an amount of the pathogen that doesn't cause death doesn't actually protect the amphibia. So when there is no protective immune response, neither in BD and B-cell. So that the problem is that we can't, for instance, do vaccination strategies. Then we can't necessarily bring out loads of amounts of antifungals in the wild to treat this antifungal diseases. However, it's really complex, because it also has to do with altitude and temperature. So both B-cell and BD have different temperature ranges in which they actually survive. And if, for instance, amphibia happen to be in areas where the temperature is a lot higher, then that can actually help. So my research personally is more really to understand the evolution of such a pathogen. And that's more in a broader context of understanding how co-evolution of host pathogen interactions actually work. And it doesn't directly translate into a treatment strategy.
Thank you. We've got a question from Alex, who says, first of all, thank you for your answer. And then has a general question asking, are frog populations finally on the recovery with the recent discovery of resilience of frogs in the Americas to BD, or is it going to get worse?
So there is some populations that have actually recovered. In terms of BD, we see some recoveries. In terms of B cell, that largely depends on how we control basically that we make sure that B cell isn't introduced into new naive populations of amphibia. Because if actually B cell gets introduced through pet trade or any other way into naive populations of amphibia, likely that will also cause die-offs if they're susceptible. And there were some trials with newts that are found in the US, that lab trials, and they were found to be highly susceptible. So if B cell would be found in the wild in the US, or it would be get there in some way, that will be deleterious. But for BD, there's some frog populations that actually are recovering, yes.
Wonderful. Well, I think that's a relatively optimistic note. If there's no more questions, we'll go on to our third and final speaker. But again, massive thank you, Theresa, for an absolutely wonderful talk.
Thank you very much.
And I'm sure you'll be available at the end for more questions should they arise.
Yes, thank you.
Wonderful. Well, thank you so much. And where's our next speaker? Oh, can we unmute? Is it Jennifer? No. Yes, Jennifer, yes, has had some tech issues and has had to rejoin, so my apologies there. Jennifer, are you able to join?
Yes, I'm so sorry. My internet just dropped out at the best possible moment.
Wonderful. Well, at least you're back now.
So sorry.
Are you able to share your screen?
Yes. Okay.
Wonderful. So this is Jennifer Dranttel from the Montfort University, and it's a very exciting talk from Mongolia.
Hi. So I just want to start by thanking everyone for speaking already. I feel like my brain is super full, but luckily for you, this is a much less technical talk now. I'm coming at you from a bit of left field, and I have a background not in biology, especially not in fungal biology. I have an undergraduate degree in architecture, a master's degree in fibers, and I'm now at Montfort University pursuing my PhD in sustainable textile technology. And the title of my PhD is Biomimicry-Inspired Material Innovation in Mongolian Gears to Combat Negative Indoor Air Quality. So that, again, is a bit out of left field. So I'm going to more kind of just tell you a story of how I came to my PhD, and I swear there'll be some mycology at the end of it. So just getting into telling you the story of it. So in August of 2018, I arrived in Ulaanbaatar, Mongolia for a 2-year teaching position without having so much as read Mongolia's Wikipedia entry. If you think that sounds crazy, you should see how I make other decisions in my life. Anyway, once I was in Mongolia, I realized there was a topic on everyone's lips about life there, air pollution. But at that point, I still didn't have a sense of how bad it was going to get, because the autumn weather was gorgeous and clear with impossibly blue skies. But the minute autumn turned to the dead of winter, freezing, brutal winter with temperatures that dropped to -40, I discovered people had actually been selling the pollution short. By mid-January, if I looked out of my classroom door, I couldn't see through the thick air to the end of the corridor. And I learned the hard way that I had to stop sleeping with my windows cracked open at night, as on several occasions I woke up to a fine layer of ash over everything in my flat and the unshakable taste of burning in my lungs for the rest of the day. As it turns out, Ulaanbaatar, Mongolia is the most polluted capital city in the world. And if I'd done any research at all, I would have seen that everyone was talking about it outside of Mongolia as well. In fact, the air pollution is so bad that the World Health Organization has declared it a humanitarian crisis. The air pollution in Mongolia is characterized by high rates of particulate matter, PM, particles, small particles of pollutants that are incredibly dangerous when inhaled. The most concerning type is PM 2.5. The particles are so fine that they pass through the lungs and directly into the bloodstream. The World Health Organization has set an acceptable level of PM 2.5 in air as 10 micrograms per cubic meter, and typical winter days in Mongolia, the readings exceed 1,000. We actually don't know how far they exceed 1,000 because the devices can only measure that high. Today, 1 in 9 deaths in Mongolia can be directly attributed to air pollution. The high rates of particulate matter pollution in Ulaanbaatar are created through burning tires, garbage, and raw coal in the poorest areas of the city called Ger districts that fan up the hillside surrounding the city center and now house 60% of Mongolia's population, nearly 1.6 million people. Ger districts are named after traditional Mongolian gers, felted wool tents stretched over an expandable wooden frame that the city's residents have brought from their formerly nomadic lives in the countryside. Many people call them yurts, but in Mongolia, the word is ger. Ger districts are unplanned and largely uncharted. They lack infrastructure, easily passable roads, and access to stable utilities such as electricity and water. And Ger districts, like similarly impoverished areas and cities all over the world, have become an easy scapegoat for all of the problems facing Mongolia. And most of the local politicians and Western designers that come in to try to improve life there have decided that the best solution is for residents to abandon living in gers and to build structured permanent homes and apartment blocks. But as a STEAM teacher and a designer, this just seemed kind of lazy to me. And I thought that there had to be a way to both preserve the connection to gers and improve life in these areas. And I thought that I should be able to come up with a design-based solution that could accomplish both. Because gers are incredible. They've been refined and perfected in practice for over 3,000 years. Each part is a wonder of engineering and purposeful intent, and they're incredibly effective shelter in a punishing natural environment. They're stable, flexible, strong, portable, and secure, and they're perfectly designed to withstand snow and wind due to the distribution of tension along the top through the side panels. They're designed to offer maximum indoor space, and they can be well heated by a single central stove. And then there's the felt. And the felt is really next level. It's fluffy and warm. It's layered on the walls in sections up to 10 inches thick. It's also used as a floor covering and for indoor carpets, cushions, and tapestries. It's made from the fibers of 5 Mongolian animals, dependent on the region and the composition of local herds: sheep, goats, camels, horses, and yak. It's naturally permeable, so it allows some airflow to prevent the buildup of carbon monoxide inside. It's naturally flame retardant and water wicking. But its importance goes beyond the practical. The wool felt is incredibly significant to Mongolian culture, and the process surrounding its making is a centuries-old ritual that connects communities and honors the spirits of ancestors who've passed down the mother felts through the generations. There's also an innate sensual comfort to being in a ger that cannot be denied. It feels cozy and inviting. It smells of a warm, friendly animal, and you feel ensconced like you're in a womb. Insomniacs all over the world agree that the best sleep you'll ever have in your life is inside a Mongolian gyre. So the problems facing Mongolia are messy. They're what designers call wicked problems. There aren't any quick fixes, just a tangle of complicated issues that seem too big to unravel and too hard to make any tangible progress on. Designers tend to only focus in on one small area of the problem, which gives them the satisfaction of feeling like they've fixed something. But meanwhile, the whole big tangle goes unresolved or even becomes worse from unexpected consequences of their short-term actions. To understand how to address complex problems, we need to be creative and we need to broaden our scope and look not just at the practical nuts and bolts issues we're trying to improve, but we need to look at the culture, at the shifting environment, at larger ebbs and flows of civilization, at historical context, and we need to project imaginative views of the future. We need to consider every possible field, idea, and issue that comes to bear on the situation, and hear the voices of not just experts, but of all those involved in the problem. And we need to then honor and privilege their lived experience. We all need to work together as co-collaborators to devise solutions to complicated problems that are highly contextualized and perfectly situated to a specific place and time. Not all citizens and regions of the world have the same agency over their role in our global environmental crisis, yet we all have the potential to take small steps in the right direction. This is a design philosophy that's still emerging. It's called transition design, and it calls on us to make actionable steps immediately to work toward a more sustainable environment and a more circular economy. The big choices we need to make, the overarching global progress, can feel unmanageable and too messy to unravel. But transition design encourages us to start thinking small and to use transitional ideas in short-term futures to make progress towards larger-term goals. Being creative in our approaches and starting today to reimagine the world around us, to develop smarter materials, work with our existing environments, and use our cultural legacies as jumping off points rather than as hindrances, will allow us to work towards a healthier planet and healthier communities. So when I began to work with my students and Gyre district residents, as well as non-governmental organizations within Mongolia, to look for design-based solutions to the problems facing them, I began to apply these ideas of transition design by looking at a broad scope of ideas and fields that can inform solutions and bring new ideas to the table. With my background in architecture and textiles, a natural first place for me to start was in material. Materials have always mattered to humans, to our culture, our mental and our physical health. And materials in a Mongolian gyre, I knew provided a really interesting springboard for further exploration. So I began a process that I call remixing. So combining materials that were already used in Mongolia and combining them and using them in different ways to add in new function and explore their potential as stopgap solutions to healthier living environments. By utilizing the natural properties that already existed within the materials and leaning into that and then adding in other elements, I thought it would be possible to create easy short-term solutions that would be adaptable and would allow residents to take quick action toward improving the indoor air quality in their gyres. So mushrooms and fungal mycelium is a wonder material that's on a lot of designers' minds at the moment, and we're only at the beginning of understanding what the material can offer us. I'm exploring its ability to combine with the wool felt already in use in gyre walls to create a mid-range building material that's still biodegradable and portable, but may lend more stability to transitional dwellings. Scientists are already proving that mycelium, through a process called mycoremediation, can remove harmful materials from soil and water. So I started to think, why not air? There are scientists conducting concurrent research around air purification potential of mycelium, and I'm also testing this premise as well in my own prototypes. So I've begun a series of experiments, which I'm now scaling up and working on how they will actually be implemented by families in Mongolia. In my first experiment, I began to see the mycelium was filling in negative space within the wool felt. And then I began to work through different ratios of colonized mycelium and wool, both loose fibers and already felted samples, to determine the best ways to create composite materials, and then starting to build a catalog of the properties of these materials. So these are just lots of weird samples of hair. It really likes the yak hair, I have to say. And basically, I also determined that the mycelium actually goes beyond just filling in the negative spaces between fibers, but it actually feeds on the keratin in the wool and colonizes the hair fibers themselves, which adds an interesting component to consider as a short-term solution, because eventually the mycelium will completely break down the hair fibers. So it could be a means to a circular economy in which it's completely biodegraded and recycled. I'm not a mycological expert like a lot of people in this room. I don't have a background in biology, but I am interested in material, and I know that there's so much potential in material development if we use biomimicry as a starting point and as a cue. So I approached my use of fungal mycelium as a designer, trying to think out of the box and look at new ways to embrace buildings, materials, and the way we think about our homes that might not occur to specialists in all of these disciplines. It's just one example of the explorations I'm pursuing and developing using materials that are already present, significant, and perfectly adapted to Mongolia. And material development is just one place that we can start as we take small steps to transition towards healthier Gare district in Mongolia and a healthier overall planet. And that's the end.
Fantastic. What a fantastically exciting talk that was, and what a way to end the night. Have we got any questions? There's lots of brilliant approach. I hope it goes well from Jane Haydecker. Karina Marker says, thank you for a very interesting talk. I'm curious, with adding mycelium, do you have any issues with spore production in the air?
Yeah, I think that is a concern, obviously. And I think while I have some beautiful little pictures, obviously, of the mycelium fruiting, which makes for a lovely picture and a compelling visual image, if you actually don't put it into fruiting conditions, then it won't fruit. So I think there are ways that it can be kept to just completely colonize, but not actually produce pins and then fruiting bodies. So it's kind of a work in progress of maintaining that. And I think the idea of this obviously is that it's a quick kind of stopgap solution. So as you know, it would be kind of a work in progress, and it would be teaching people in Mongolia that this is something that can be applied during the felting process and then can go into their homes. And so, you know, with that, there's going to have to be some education around like certain conditions. But I think there's potential there. So that is the part that's still slightly unresolved.
Thank you. We've got several questions have come in now, and they look good ones. The first is from Karina, who asks, do people in the Gare districts live there all year round, or do some people migrate to the city only in the winter? I was lucky enough to spend a month in Mongolia a couple of years ago. I love that you're trying to enable people to stay in their gears, as it's such an important part of their nomadic lifestyle.
So there are definitely still true nomads in Mongolia, but at this point, it's only about 30% of the population. So most people that live in the Gare district are living there permanently. And there have been several major waves of kind of rural to urban migration that has brought waves of people into these Gare districts. The first was in the 90s with the end of communism in Mongolia. And recently, there have been a lot of waves due to climate change. So as people are losing the ability to actually sustain themselves in a nomadic lifestyle, due to climate change, economics, family dynamics, whatever the reason, they're basically economically destitute, and all they have left is the family ger, and they bring that and set it up just kind of on bare land in these like rapidly growing districts. So they are living there all year round, and the winter is the main issue, because that is when people are burning anything they can get their hands on to stay warm in these freezing temperatures. And that's where you really see the problem of the air pollution.
Excellent answer, and a perfect answer for our next question from Kai Cartwright, who asks, I'm really interested in how you're testing the ability of the material to absorb particulate pollution. And also adds, thanks for the great talk and good luck with this great project.
So that is a bit, again, of kind of a work in progress. So in two ways, I have a test ger in Mongolia that I have some prototypes set up in, and that is balanced against there's a pollution reader in that ger, and it's being operated completely normally with a coal burning stove. Just the only difference is the addition of these prototypes. Then that's against kind of the readings that are in several other gers in Mongolia that are not doing any interventions. And as well, there's a company that we've, through my university, we've sent several of the prototypes off to, that are basically creating kind of a polluted air chamber and then testing the levels of the pollution as the mycelium is within that chamber. So kind of like in practice and also in a lab.
Brilliant. And Kai says, oh, that's awesome. So fantastic answer there.
Good.
We've got a question from Polly Bates, who says, as an artist also interested in how mycology can reverse anthropogenic effects, it's great to see fellow creatives take an interest in the possibilities. How did you arrive at using the pink oyster mycelium?
Well, if I'm honest, actually, when I was getting my master's in textiles, I did a lot of really strange textile experiments, and kind of through an accident of having a mushroom grow kit stuck in the bottom of a cupboard for years, and then discovering that it actually had grown through everything else I had in that cupboard, I started growing mushrooms through silk fibers. And so I used pink oysters and just various, not just pinks, I like pink, but using oyster mushrooms, which are really easy to grow, and they'll grow on a lot of different substrates. So that was kind of when I first started thinking about this problem in Mongolia. I had lots of different ideas, and actually fungal mycelium was not on the top of my list. I really envisioned I was going to have some sort of cool lichen situation happening, but that was kind of not moving quickly enough, as lichen does. It takes generations to grow, and I kind of lost the will to carry on with those experiments. And so I started thinking, well, you know, I know mushrooms will grow on anything, and I bet they'll grow on wool, and kind of understood how they worked and started really looking into that as a potential, and then it just kind of grew from there. So reishi have the fastest growing and the thickest and strongest mycelium, and that's why a lot of like myco products that you see now use that type of mushroom, and that is something that is native to Mongolia, so that is one I'm exploring as well. But for me, the oyster was kind of like a first easy jumping off point of how to just play around with this idea initially.
Brilliant. Thank you so much, and thank you again for an absolutely wonderful talk. At this point, I'm going to ask if Theresa and Chris can come back as well, and we should be able to spotlight everyone. Add spotlights. That's brilliant. So at this point, any questions for any of our three wonderful speakers? You can ask it to one of them or to all of them. And it's really, yeah, any questions you like. Suppose we're waiting for, this is to everyone. How has it been doing a PhD during a pandemic? Particularly such active ones.
Probably a bit more of a challenge than a normal PhD, I would say. So yeah, I'm quite lucky that I just work with data, so I didn't have the whole lab problem that I couldn't access a lab. But yeah, I mean, it has been a challenging time for everyone, right? So.
Yeah, so we were quite lucky because we do like food science. We counted as a, like an important company, whatever. So we kept labs open all the way through COVID and stuff. So other than it being a little bit quieter around and having to social distance and wear masks, it was probably all right. But yeah, I think I was quite lucky in that respect.
I was actually living in Mongolia when the pandemic started, and thought it was just going to be like a little Asian blip, and packed an overnight bag as I was teaching and my school was shut. We were teaching remotely. So I just packed an overnight bag and came to the UK to spend what I thought would be a couple weeks with my partner. And oops, 2 years later, I'm still here. I got stuck and then started my PhD. So I had the idea, cooking, and I had a proposal written, but I wasn't planning to start, as I still was planning on teaching a couple more years in Mongolia. So yeah, it kind of just sped it up. And then I spent the first year just alone in my bedroom reading and growing mushrooms, which I think probably a lot of people did during the pandemic.
Fantastic. So we've got one question for Chris. Oh, I presume for Chris from Cameron, who asks, I have apple trees in my garden that may need to come out. What would you plant instead?
Well, in terms of if you've only got a couple of apple trees in your garden and you just want to get a few apples a year to eat, you'll be fine with planting another apple tree in the same place. You probably won't notice the difference. It will still fill your fruit bowl every year. It's always one is they go, I've got two trees in my garden. How do I like deal with these trees? And it's like you don't want more than like 2 trees, otherwise you will have the whole neighborhood coming around looking for apples. But yeah, instead, I mean, if you were to plant something else in your garden, you could just move the tree slightly, but you'd be fine. Like with any type of apple tree, pretty much you'd be fine.
Oh, wonderful. He says, cheers, Chris. So some good news there. Joni asks, and firstly, what wonderful talks, what fantastic mycologists you are. We're very excited. But Joni asks, were you guys always interested in mycology or just when you began your PhD? And should we start with Theresa to Chris to Jennifer?
Not always. So I started out in the very beginning actually wanting to do neuroscience, then switched to biochemistry, then switched to toxicology, then ecology, and ended up in medical mycology. So I think I'm broadly interested in life science, and I find topics that I get into fascinating. But for me, it actually was somebody I met at the conference, and Dr. Alistair Brown, who will ask one question, and he talked about this PhD program, and I found it really interesting, and I thought, okay, why not? And now I'm really intrigued.
So yeah, I did my final year project at NIAB. So obviously I got exposed to horticulture quite quickly, and mycology and horticulture pretty much go hand in hand if you have anything to do with pathology. So it was quite an easy transition for me straight out of uni. So yeah.
Yeah, I mean, I would say just as much as kind of a novice, but then kind of from a materials and a designer standpoint, it's really I worked a lot in fashion in my master's program. There's a lot of talk around myco leather at the moment. And then again, within architecture, it's used as a building material. It's kind of a hot material at the moment. So I think I kind of came to mycology like from both ends of just being kind of a random nerd that spent a lot of time in forests when I was young, to then kind of thinking about like, oh, actually this has some other applications that are really cool.
Wonderful. We've got a question from Polly Bates to Chris and Theresa, but Jennifer, feel free to chime in as well if you have an answer, which says, how important is mycology in the research and reaction to climate change? And how has it, and I'm assuming it is climate change, affected your particular study and results? Should we start with Chris, then to Theresa?
Yeah, so I have a paper submitted at the moment about climate change effect on apple soils. So hopefully that will come out in the next couple of months. And it basically describes the effect of sort of increased CO2 and temperature effects on the soil microbiome. And what we tend to see is that the management effect is more important. So we tested organic soils versus chemical applications into soils. And what you tend to see is that's the most influential factor, as you imagine, with things like fungicides and everything applied there. That has the most impact. Climate effect does have an impact, not so much on fungi, more on bacteria. We tend to see that that's where most of the influence is. But yeah, so there is some sort of limited effect there. How much it will be in future, we'll have to wait and see. But soils are quite complicated because climate conditions change a lot in soils compared to environment. So as you go deeper, things like CO2 concentration go up a lot faster. So there's obviously a different impact factor there. But a lot of the stuff we find in soils are already tolerant because it's higher. So we'll see. I think temperature is the most important. And extreme weather events, flooding and stuff is going to be way more of a factor than things like temperature and CO2.
Yeah, just in general in medical mycology, so many human fungal pathogens are opportunistic pathogens, and obviously we have a body temperature of 37 degrees roughly, and that when we increase that, it is not ideal for most fungals, for fungal pathogens. For most fungal species, because they're not adapted to that. When the overall climate, the overall temperature increases, there is a higher risk that there will actually be fungi that are adapted to higher temperatures, like our body temperature. And then we have more opportunistic fungal infections, or we could have opportunistic fungal infections. And it has been proposed that this has also something to do with Candida auris outbreak. Because but that is not my own research. So in context to myself, to my own research, so I mean, climate change exerts a pressure on biodiversity anyway. Having Chytridiomycosis around is yet another factor that can reduce biodiversity. So in that context, it exacerbates that. And as an environmental stressor, it can also it could hypothetically have effects on transposable element proliferation, because they have been observed, so expansions of transposable elements have been observed when there's environmental stressors increasing. So it might be something that also plays an effect at some point. So, yeah.
And we have a question for Jennifer from Stuart Skeets, who asks, I have seen mycelium used in cavity walls, but have you ever seen it used as a mycelial layer on the internal side of an external wall for heat conservation?
Yeah, I think there's a lot of companies now that are coming. I feel like we're still at kind of the beginning of this uptick. I think in another 20 years, you're going to be hard pressed to find a material that's not involved with mycelium. But I think there are companies that are using it in cladding, in panels, things like that. I thought it also was kind of particularly suitable for Mongolia because in general, people living in a ger have a different perception of what a building is and what a home is already. And so they're much more open to a more portable, already it's a portable structure, but to something that wouldn't feel as stable as, you know, we build with brick and wood and, you know, steel like supports, and that is not something that registers to them as necessary. So I thought it was kind of a really good bridge for introducing that as a building material. There's almost like no issue for them kind of conceptually to take that on board.
Wonderful. And I think following that, I've got a question that's been sent to me privately from the BMS office. And there's a typo, but I'm going to read it with the typo because it's quite a fun one. And it says, if the fungal mycelia eventually breaks down the hair in the ger, would it need to patching every so often? And then is it time to save a yak?
Well, yaks are interesting. Actually, they are, yeah, endangered, not for any reason other than they breed prolifically with the cattle. And so it's really hard to keep them separated. So most yak and cattle in Mongolia are like a genetic combination of the two, actually, in this day and age. Yeah, trust me when I say I know way too much about yak hair. So yeah, I think to go back to the actual question there, yes, so eventually it would degrade the wool, which also is kind of an interesting idea of how the wool works in Mongolia. So every fall, a new layer is felted onto the existing layer. So that is why, like in some gers, in some places, the wool is like 10 to 12 inches thick even. And they call the existing felts the mother felts. They believe that those hold the spirits of their mothers and their female ancestors that pass down the gers matrilinearly. And so every fall when they shear the animals, they lay out all the felts that are already used in their gers, and they felt a new layer on top of that. So within that process, I thought that was an easy way to introduce mycelium spores. And then in the following season, a new layer can be felted on top of that.
Wonderful. And again, another question from the BMS office asking, could the fruiting bodies in the ger be a food source?
Yeah, I think there's definitely potential for that. I think it's a bit I've not found a lot of research around people doing any studies about, you know, when you think of people that have done studies around mycoremediation, like removing oil spills, those fruiting bodies were not used as a food source. So there would be some, you really need to test that specifically to be sure that if it is actually removing like heavy metal pollutants from the air, or from like settled pollutants from the pollution, that it's still safe to eat. So there would be a little bit of concern around that.
Wonderful. And I believe we've now dried up some questions. So I'd like everyone to join me in giving a massive thank you to our three wonderful speakers, to Chris, to Theresa, to Jennifer. Absolutely wonderful, it was absolutely brilliant. And then finally, in reverse speaker order, so starting with Jennifer, then Theresa, then Chris, it's a quick, any sentence you want to end on, any take-home notes for anyone to take with them into this Wednesday evening.
Oh, yeah, I just want to thank you for letting me speak here as someone that feels wildly unqualified to be in this room, but as someone that's really interested in the kind of merging between science and art and science and design, and just using those intersecting points as places where we can sometimes find new voices and find interesting solutions by kind of thinking out of the box a bit.
from my side, just very much thank you for giving me the opportunity to speak here. And it was really interesting to listen to both you, Chris and Jennifer. Really enjoyed your talks. Yeah, thank you for that opportunity, and thank you for the talks.
Yeah, just to echo them, thank you very much for coming. And if you get a chance to, just raise a glass of cider and support the cider industry. I'm sure they'll appreciate the plug, because we need to make sure we're supporting the cider industry, because they're having a lot of problems at the moment. So we don't want to lose our cider industry. So yeah, make sure you buy local and buy good quality cider.